Hi, my name is Nick, and I am in the Onufriev lab, and I'm co-advised by Dr. Igor Sharkov, and he works in a mosquito lab. So in some lower eukaryotes, special cells replicate their DNA many times without cell division, and consequently the chromosomes become highly multi-stranded and visible in a light microscope. So that's what you see right here. That's a light microscope picture of some chromosomes. These are called polytene chromosomes, and they're useful for studying the 3D organization of chromosomes in the nucleus. Recent studies have also shown that there is a correspondence between these polytene chromosomes and their non-polytene counterparts. Experimental reconstructions of these nuclei have identified 15 positions along these chromosome fibers that are frequently attached to the nuclear envelope. So we built a computational model to address and answer 3 questions. First, if you place polymers in a highly confined spherical container, like a nucleus, are certain positions going to find themselves up against the nuclear envelope very frequently just due to the confinement in this very specific geometry? And if so, do these contacts correspond to the same 15 contacts that were observed in experiments? So is confining these polymers in this geometric way enough to account for the 15 attachments that they observed in experiments? If not, you might assume that these positions have some intrinsic affinity for the nuclear envelope. Second question, how likely is it that any of the experimental attachments that they saw corresponding to the peaks in this red line here, how likely is it that any of those attachments were observed just due to chance? And we answer that question also, because chromosome folding is not like protein folding. It's not deterministic. It's stochastic. Chromosomes fold in non-random ways, but not the same way every single time like a lot of proteins do. And then the third question that we answer is, if you build models with and without these chromosome nuclear envelope attachments, how are you going to change the overall 3D organization of the genome? And that's summarized by these blue matrices. And you can see that when we start adding chromosome nuclear envelope attachments in the positions that we identify with the assistance of our model, you get something that more closely resembles experiment. And this is the most current results that I have, and you can come to my poster.